Number 7.
Number 7, okay, got it. All right. So the thermocycler breaks down. So let's talk about the thermocycler again, because we actually had an issue with the thermocycler this last fall, and that's why some of our PCR results didn't work on the row one in the block didn't actually work for this exact reason. So thermocycler breaks down. So thermocycler is heat and cool, right? So the annealing process and denaturing process, that's what happens. So it heats it up, things break apart, it lets it cool down, things run, and they elongate. And you find this, right? So they're broken apart, but they are not elongating. So what step was broken?
After cleavage?
And we know which step of the process of this is actually broken. The annealing part, right? We're not actually elongating. So that's broken. So immediately after, which of the following processes of the thermocycler break down? Well, it did not elongate. So the primers have binded, but DNA polymerase has not been able to bind and elongate. Okay, how do we feel?
Okay, thank you.
So even though we didn't do the PCR worksheet, PCR is a great review of DNA synthesis, because DNA synthesis is literally you copying DNA. PCR is we are copying a component of DNA. So we are trying to replicate that one part over and over and over and over again. And our target sequence is that between the two primers, right? So an idea is just to amplify it to such an extent that you can physically see it in a gel. That is our goal of PCR. So I highly recommend going back over and looking at PCR, just because it really does kind of recap DNA replication really well. All right. What other questions do people have?
Oh my God. Blackjack Beauty. So cute.
Yeah.
Oh my God. You should just
No questions?
type in the chat. I don't want to speak.
How do you genetically reproduce? I'm just kidding. Genetically.
It was like I had my one question. I'm done. We're good.
Or you can unmute? No.
Ray can unplug. Speeding.
What?
Get on there.
They're what pedigree that?
Ricky, I have a really random question.
Okay.
Do you know off the top of your head what kind of you go ahead. What kind of microscopes we have in the lab?
I believe we have Nikons.
Okay, cool, thanks.
That we have the department either have Nikons or Leicas, depending on which classroom we're in. I believe ours is Nikons.
Okay, you have microscopes. You have your own personal microscopes, right?
I do. Yep.
What do you think is the best kind of microscope?
That's a hard question. So microscopes range in price from like $20 to like $50,000, depending on what you want.
Mm-hmm.
The microscope in our labs are about $4,000 a piece.
Okay.
So like they range in price a lot. I have a field microscope that I take in the field with me, and it's like $500, and it is like awesome. It goes to 40x. The visualizations are okay, but what's neat is it has a camera actually built into it.
Okay. Cool.
That's kind of neat. Charlotte and I were just gifted, portable microscopes that attach to phones. They were at her work giving them away. So like, we haven't tried it out yet, but there's all kinds of microscopes.
That's so cool.
Okay, cool. I was just, the only reason I'm asking is because we have a really, really crappy one at home, and after yesterday's lab, I was like, I mean, I'm not gonna go buy one right now, but sometime I would really like to have a better microscope, so that's all.
So I'm going to give you so all of you don't do this because you're gonna go bankrupt. There's a thing called surplus. So at the state level, anytime we want to get rid of something, we have to surplus it. And to surplus it means they sell it. So UW has a surplus store, Everett Community College has a surplus store. So if we have a microscope that we're just done with, we will surplus them. So let us know, because we often have surplus microscopes, like things that we just rotate out. Or let's say the 100 lens gets broken and we can't fix it. So you have everything else. You're never going to use the 100X. You can buy them for like $2-3.
What?
Oh my goodness.
Background.
Computers from the university and college, you can buy for like $5-10. TV screens for like $5-10. You can buy cars. So like anytime we get rid of a car or a van or equipment, it's like $50 to $100 to a couple $100. So I highly recommend, if you're wanting to make a big purchase in your life, look at state-sponsored surplus stores first.
Okay, I will look around. Thank you.
And if you see one, get me one. I'll pay you double.
I have a question about the study questions.
Okay, which one?
11A, I believe.
On which study set?
I think it's the 9.
And we're gonna have to tell him.
He's going off for a second.
Did you need me to turn this
Okay,
off? Okay.
more amplification questions. Share. This question?
Yes.
Okay.
How do we know how long the PCR would be? I'm a little bit confused by the question, I guess.
So we need to design, okay, if our target is those sequences, we need to design a target to capture that, right? And primers, how primers work and PCR works is you bind to the primer, just like you do with initiator proteins or in DNA synthesis. We have the primers, right? And so that primer is where the polymerase, DNA polymerase, is going to bind to. So if you have a very specific sequence in mind, you choose the nucleotides in front of it that will give you that exact sequence. Okay? So we have this sequence, right? And we go 5' to 3', we're going to build on the 3' side. So we need opposite of that. So you're going to have a leading template primer and a lagging one, okay? So they're going to go in each direction. So every PCR has 2 primers in it, 2 sets of primers. And then there you want it to encapsulate the whole sequence. So for instance, if we're going to build the bottom string, the primer is going to be complementary to the top and it'll be complementary to the right-hand side. And you need to be at least 10 nucleotides long because you want your sequence to be a sequence, like something that's not going to be repeating in your DNA. Because if you have a DNA that's 10 billion pairs nucleotides long, and you pick only 3 nucleotides in a row, well, your primer is weak. It's going to bind everywhere. So you want something highly, highly specific. So that will be the primer that binds, so complementary to the top on the right-hand side, or complementary to the bottom on the left-hand side.
Okay, so for question B, you could pick any amount, or does it have to be specific?
How long would the PCR product be? So then you calculate, well, okay, so our primers are, say we do 10, then we count out how many nucleotides we have left within that bubble.
So because there's 5 primers,
5 primers.
before the box, okay.
Oh, no. So you do it in the box. So the PCR will be part of that, most likely.
Okay. Okay. Thank you.
I'll let him know that he's got
So remember, how does DNA replication happen? We open up our bubble, and then, so we break DNA apart, right? So this is the degradation phase. This is in the thermocycler, this is the denaturing phase. Everything breaks apart, falls apart. Then we put the primers bind to specific locations, and then polymerase runs and goes to the end of that bubble. That's exactly what we're doing here. It's just instead of using all of the other initiator proteins and helices and all those, helicase and all that, all we're doing is we use heat.
I feel like we shouldn't even process so anything for PCR.
You have a question on your message?
On the pedigree lab. Is this the MAMO 2 question? I don't have it up yet.
I think so. Where is it?
It's the linkage question.
Do you want to say something?
Sure. No, it's the question about the linkage.
Got it. Okay. So, first things first. All right, let's talk about MAMO 2 and then we'll talk about this first. This is Boris's genotype. This is his karyotype map. Are there any abnormalities in Boris's karyotype map?
Yes. I don't know that I can shake my head this far, but you can tell.
Anyone?
Yes.
So any abnormalities? What's the abnormality?
What is what?
The abnormality. What's wrong with Boris's karyotype map?
Wasn't it the X chromosome?
Is it the X chromosome?
Okay, what about it?
It wasn't there. We did
It's
that
not there.
yesterday.
Excellent. So we're missing an X chromosome and we're missing, or we have too many?
21. Too many 21 chromosomes.
Too many 21s, and what about 16?
Not enough.
Not enough. This poor kid's really just screwed up. Okay. Alright, hold on 1 second. I didn't open it. There we go. Okay. So then let's talk about MAMO 2. So, because I keep getting lots of questions on MAMO 2. And this will go to understanding all of it. All right, how we read this? The gels are the individuals. Individual 1-1, 1-2. So this is it. Fred is individual 1-1. Ethel's individual 1-2. Generation 1, individual 2. Okay, so that's how they read. So individual generation 2, 1, 3, so forth. MAMO 2. So if you have these specific bands, you have MAMO 2. Of these individuals, who has MAMO 2?
I1.
2-1 is exactly the same as MAMO 2. So that individual has MAMO 2.
Oh, okay.
All right. Then who?
-6,
right there, 2-6 and
3-1.
3-1. Okay, so then all you do for the memo 2 is you fill in who has what. So what was it? 1, 6, and 3-1, Boris?
So you put
Okay.
memo 2 there?
So memo 2, then we figured out memo 2. Then we go through and you gotta figure out, is any of these X-linked, right? So Boris doesn't have an X chromosome, so clearly something can't be X-linked. So that does help us. So then the question down here becomes, does this linkage of information clarify Boris' genotype at any loci? What do you guys think?
Well, it should, right? With the
Any
if it
ideas?
matches, no? Or the link?
I don't know.
I say yeah, because the loci indicates that he has it.
He has it. Okay, excellent. So it's not X-linked. Does anybody know what MAMO2 is? Anybody do any research on MAMO2?
That's right. Oh my god, I'm white. I tried, but I
MAMO2 is the breast cancer
just
gene.
these questions.
Oh.
Okay. So it's not X-linked. All right. So we know that. What else do we know?
Mans has a Y chromosome.
You want to say it? I'll say it.
He has a Y chromosome.
Has a white chromosome?
He has a Y chromosome, but not everyone in this phenotype map has a Y chromosome.
Okay.
So can we actually tell what chromosome MAMO2 is on? It's not associated with blood type, not associated with Rh factor. It's not associated with color blindness, because that's on X, or hemophilia. So we know it's not associated with any of those, but we don't know what it's associated with. So yes, you can clarify that some of the linkages we can eliminate, but we cannot tell you which one it is.
Okay.
That's the answer to that question. Next question.
I guess maybe briefly go over this question 5 on the same pedigree lab.
Question 5. Okay. Okay. Give me one. So this is a Punnett square. Okay, so let's do, so Wei Sheng's genotype for, he's IA, so we're A and B. It doesn't even work on this computer. And Adele's, she's A and O. Okay, that's the two genes. So, oh my goodness, this stupid computer. and then so we have the little I, right? So what's the probability of getting AB?
Just like 1/4 of the time.
1/4, excellent. So 1 out of 4,
Oh, that was,
okay? And then you have to do the same gene, you have to do a different Punnett square. It wants me every time, okay? What he's showing is RR, so he only has 1. So it's a 1 by 2, because we have big R and little r, okay? Oh my goodness. This computer's not touchscreen, so it's way harder. Okay, so we have big R and little r, so
Somehow.
what's the likelihood of having negative blood type?
I'm being recorded.
1/2, right? So 1/4 times 1/2 is what's 1/4 times 1/2?
1/8.
1/8. What's the likelihood of Boris's blood type? Or sorry, Wei Cheng's blood type. 1/8, right? So you just have to multiply the Punnett squares together.
oh,
Because they're two independent Punnett squares, because they're two different genes that are not linked. So you just multiply the Punnett squares together.
alright. Got it, thank you.
Do we have any other questions?
Any other questions?
I don't care if it's not even 100% correct. The last
I guess for like the lab we did yesterday, just for just
try to
like
take
the
everything.
second, just common question, this is just related to like the alcohol and nicotine.
What's the question say? I don't remember.
It's just how can you interrupt neurulation without basically removing any cells?
So yeah, so you're inserting something that's interrupting the signal, you disrupt stuff. So yeah, so ways you can interrupt the process of neurulation without physically removing any cells. So we stop noggin, interrupt BMP4, we change the receptor. You could have things move too quickly, too slowly, DNA replication be changed, transcription changes, any number of things.
Okay.
So what's alcohol doing in these eggs?
It's preventing the growth.
Yeah, so it's changing stuff, right? So what is alcohol? Alcohol is an OH group. It's changing the metabolic pathways. It's changing how we bind to things. It's probably changing transcription and regulation. So it's affecting all of those pathways.
Are we allowed to ask a question about the practice exam? Cause there's something here.
I typically don't like doing that because people haven't done it and I don't want to change their. Yeah, we can try to do something similar. What's the question? Which
Well,
question?
one of them just says which cells are at the same stage labeled G, large cell labeled G in the drawing, but I don't see any G, and I don't know if I'm going crazy, but there's nothing labeled G. It's for number 4. Maybe it's pointing down the air.
I gotta open it. It'll take a second. It's not wanting to open. God, I hate Canvas. All right, which question was it?
Number 4 and 4C.
Oh shit, yeah. I think it got cut off in the printing.
Okay. Is it just what that arrow's pointing to?
Yes, it's just what the
Okay.
arrow's pointing to. That's G.
Okay.
This is literally copied from your guys' worksheet.
Okay. I see.
Yeah,
Yeah.
so key, it makes it really obvious. I'm sorry. Yeah, it got cut off in printing. I remember this now.
Okay. That's all I need to know. I was worried I was maybe going crazy, so thank you.
Yeah, we didn't solve that happening. We just solved this one.
Okay.
See, this is where you can't do this on Zoom, because that was funny, in my opinion. Because I'm going crazy every day.
I have a question for problem set 9.
Okay,
It's number 15. It says, could this be a dominant trait? So from what it looks like, the trait's being passed down to its children and then grandchildren, but then 2 of its children, it skips them.
okay.
If realistically between like that group, it looks like 80% of them like have the trait. So would it still be considered dominant if it's just skipped a generation?
Which question again?
15.
That question?
Yeah.
Alright, so it's in 1, and then it shows up in 3 of 4 children. So that's probably dominant. And then it shows up in both children. So that's most likely dominant.
Okay, thank you.
If you ever thought they're not monkeys, they are definitely monkeys. He's over here just crawling and changing and yapping and talking and reaching for bottles and being crazy. She's adorable though, but she's crazy. Alright, next question. No questions? I'm down to go have breakfast, so.
Just want to clarify something on, I guess, the other, virtual lab, the Sea Urchins.
Uh-huh.
When we were like asked during like neuron development for like, when asked for like what type of neuron,
Mm-hmm.
are supposed to have like an actual like location where it's from, or it's just like type T, that's like how it's,
Yep, just what you have in the simulation.
okay, yeah, thank you.
It's kind of a neat simulation. You can kind of see how the neurons work. And this is going to be exactly what we start with next quarter when we start thinking about action potentials in neurons.
I have a question about number 10 on the study questions.
Which packet?
The 9. Study questions 9, I think.
Still the same study questions. Okay, question 10 you said?
Yes. So, for A, it would just be them drawn on the same chromosome, and B, them drawn on a different one. That's it, right?
Diagram between all possible metaphase 1 minus females, assuming they are on different chromosomes because they're independent assortment. Yep, okay. And then draw a diagram on the same chromosome that says dependent assortment. So there's two different Punnett squared possibilities, dependent and independent assortment. Yes, so this is exactly what we did second week of 221.
Okay. Okay, thank you.
For study questions, we have 9 and 10.
All right, any other questions?
I have no more questions.
Oh, she said no more.
And we can always do that. We're going to do this again on Sunday night. And you guys can message me. I'm around all weekend, so nothing's going on this weekend. So if you guys have questions. She's discovered how to scoot, and so now everything is moving around. And she's just like, I'm monkey. All right, so we call it? All right, guys, I'll see you Sunday night.
Getting a little fun.
Yes.
There we go.
I'm
I thought
gonna.
someone was trying to say something wrong.